So welcome to marine biology. I'm so sorry that you can't see me. I'm actually wearing one of my fiddler crab shirts for you. So it's unfortunate that you can't see me, but hopefully again, you at least were able to attend the online orientation. So you got to see me in person there. So anyway, I apologize. Screencast-O-Matic was just not wanting to work that function for me. So hopefully I will get it worked out for future videos so you can see me on and off in the video. So welcome to marine biology again. One of the first things I always like to start off with, these are just a few images that I've taken. I am an avid scuba diver. I'm sure you already know that from the orientation. So these are just a few of the images that I've taken, and I'll try to incorporate them throughout the semester. I love to share stories about the places that I've been lucky enough to dive at, and so I, of course, love to share those stories about all of these marine organisms with you. You'll find that I'm very passionate about marine biology. It's one of my favorite courses to teach, and it'll be nice once all the courses are back in person and you can see me in person again. So what is marine biology? Okay. Marine biology very simply is the scientific study of organisms that live in the sea. And so you may ask, all right, well, I know I need to take a biology. It's important to take, some type of science course for my prerequisites, but why marine biology? Why, here we are, right, in the middle of the desert in Arizona. Why is marine biology important to you if you are a business major, or if you are a computer science major, or whatever field of study you're in? Why would marine biology be offered, right? And so that's always the big question. So why study marine biology? And there are a lot of really good reasons why you should study marine biology. One of the really simple practical reasons is that marine life is just kind of this vast source of human wealth. You're going to find that it's in a lot of our foods, even just there are certain food derivatives that are in almost every food that we eat. And those food derivatives are going to come from marine organisms. There are medicines that we have that are derived from marine organisms. Also, we're doing all kinds of, there's medical advances in terms of using marine organisms for some of these medical advances. So for example, they're doing studies of animal immune systems in sea anemones and sea star larvae. They're also doing some research. They were doing it over at ASU on coral, because what they found is, interestingly, coral don't have what's called neoplastic growth. They don't have tumors. They don't grow these abnormal growths. And so we want to know why. And if there's some chemical that prevents that, that's very important, because we would like to potentially use that as a treatment for cancer. So again, there's many resources that we derive from marine organisms. There's many raw materials, as we're going to get into in just a minute when we talk about things like carrageenan and things like that. And then tourism. So we have this huge industry, right, that's for tourism and recreation that's built around oceans, basically. So again, you can go scuba diving, you can go hang out at the beach, you can do resorts where you can go boating and fishing, and all kinds of things. So when you talk about the economic importance of, and I think I have that down here, the economic value of our oceans, we're talking about in the trillions of dollars, so about $20 trillion a year. So, if you're in business and you're like, well, why do I need to study marine biology? $20 trillion a year is quite a bit. So there's a huge economic value in our oceans. As we kind of go backwards here, another very fundamental reason why marine life is so important to us, and oceans in general are important to us, is that about 50% of Earth's oxygen comes from oceans. So believe it or not, a huge chunk of the oxygen that we have is actually made by primary producers in the ocean, especially plankton. So again, that's another big reason why we want to study, right? The oceans. Also, oceans cover about 71% of our planet. So oceans are really important in terms of regulating the Earth's climate. And this becomes really, really important in terms of what we're seeing now with, talking about climate change and seeing an increase in hurricanes and tropical storms. So it's becoming, vastly more important for us to understand what's happening in our oceans because that impacts us directly on land as well. We talked about the economic value. Also, again, simply marine organisms are a great window to our past. So we know that marine life originated in our ocean, or we know that life in general originated in our oceans. So we're going to talk quite a bit about this as we go through the marine invertebrates. And it's a really interesting window to our past, to the origins of life. And then preservation of diversity. So again, we've talked about all of these resources, right? And I'm just going to go ahead and use my pointer here. But all of these resources that we use, foods, medicines, raw materials, that come from the ocean. So the preservation of that diversity is really important. And especially when you're talking about certain chemicals that could be derived from marine organisms to provide medicines for treatments. This becomes really important. So as we're seeing in a time period where we're seeing losses of biodiversity, unfortunately, we see that loss of things like chemical diversity as well. And that loss of chemical diversity, it can mean loss of medicines, loss of treatments, potential treatments for us as well. So preservation of that diversity and biodiversity isn't just important, because as, biologists, we want to, have that biodiversity because we want the organisms on the planet. We want them out there to study and to look at and observe. But it's also important to us in terms of our own survival as a human race. So again, just to give you, that just hopefully gives you a little idea of why you might take marine biology as a student, as a business student, way out here in the middle of the desert. So just kind of for fun, to give you an idea of all of the products that you may use that have some type of connection to a marine organism or something derived from the ocean. These are all the products. I mean, anything from baby food, beer, breakfast bars, cake mix, chocolate, milk, cookies, dog food. I mean, you can read the whole list. Lipstick, right? So you can see in this picture here, in this image, they tried to put as many of these products as they could in there that are derived from our oceans. And you can see there's a vast number of products that we use that have some type of connection to the ocean. One of the most common ingredients that you have probably heard about these days is carrageenan. It's a very simple polysaccharide that's extracted from mostly red algae, seaweeds. We call them seaweeds, but as you're going to find out, seaweed is going to be any type of marine macroalgae, and some of them are brown, some of them are green, and some of them are red. So carrageenan comes from our red algae, our red seaweeds. Different red algae species will produce different types of carrageenan with different properties. And so let me just turn this pointer off real quick here. You can go to this link here, and I have it, and there's a quick YouTube video here that, unfortunately, is going to have an ad, so let me skip it here. You can see some of these products derived from carrageenan. So again, carrageenan is this polysaccharide extracted from certain red seaweeds. And there are different types of carrageenan depending upon which red algae you use to derive it. So here's some of your uses of carrageenan. So it can serve as a stabilizer, can provide texture for certain products. It induces, because of freeze thaw, stability. You guys can read all this. Ice cream, of course, very important, reason for carrageenan there. So again, body and consistency, that's a big use for carrageenan. Sliceability, very important for your cheese. Ah, there you go. Beer improves shelf life. It's super important, right? For our lives these days. Dog food. So again, you can kind of get the point here, but these just kind of a fun quick video for you on all of the different major uses for carrageenan. So you're going to see that carrageenan is used in many different products. Again, here's carrageenan everywhere. So this just shows you, it's a quick slide. Again, you don't need to know all of this in terms of what carrageenan provides, but I just thought it would be interesting for you since you'll hear about carrageenan these days a lot more than you used to before. And it's in almost everything. So look at the back of your products tonight when you're using some of them and look to see if carrageenan's in them. If it's not carrageenan, look to see if they have one of these other two products, alginate or agar. So you can, alginate comes from brown algae, agar comes from red algae. And so you can look to see, and you can see the use of these products is pretty huge. And it's interesting because Chemical and Engineering News came out with an article. You can click to the link. I'm not going to open it here for you today, but just kind of fun if you are in the business field. An interesting article that talks about the industry based around seaweed. I mean, it's a multi-billion-dollar industry, really interesting. And one of the reasons for it is that it's just very easy to cultivate. If you look at seaweeds, very easy to cultivate, and it's natural in origin, which is important because consumers these days are demanding more bio-derived products. So anyway, you can read about this, but it talks about the actual industry of seaweed and the fact that it's valued at more than $6 billion, which is amazing to me. So seaweed is where the money's at in terms of marine organisms these days. There are other animals that produce some raw materials for new drugs. So again, I'm not going to go through this slide. I'm not going to necessarily test you on what all these raw materials are. It's just to show you that marine organisms are being used to produce raw materials for things like new drugs. So you can kind of go through and read some of these. They're really interesting, like tunicates. Produce a chemical that's being tested in humans for treatment of breast and ovarian cancers. We talked about, again, corals are being tested because they don't, corals don't have neoplastic growth, and they want to try to identify what chemical prevents that. So again, just some really interesting uses of these marine animals for potential new treatments. So this is why you're taking marine biology. Another reason why you are taking marine biology is simply just to learn a science. And science is an important way of knowing, is kind of how we initially put it, because it's a science. And the nature of science is this kind of process of inquiry that is going to include repeatable observations and testable hypotheses. And this is important because this becomes really exponentially more important to us now in a world where information is at our fingertips, so readily available. And some of that information is good information, and some of that information is really not, really bad information, right? And we've found this to be really evident with the pandemic lately in terms of finding information on that. So it's important to understand the nature of science and how we do science and why we do it this way and what value it provides us. So let's talk about the two different kind of methods of science. There's what we call discovery science, which is using the process of induction. And this is where we make observations. We are going to collect our data. And oftentimes, a lot of times that data collection is done out in the field, but it can be done in a lab as well. And then we'll make up a general hypothesis based on that discovery science. Now, most of the time, discovery swhich as I use my pointer here, discovery science is going to lead to the hypothetico-deductive model. So which is going to use the process of deduction. And so that's going to be the process that you probably learned about, where we actually use the scientific method, this very organized way of doing, this organized method of doing science, where we come up with a hypothesis, we come up with a prediction based off of that. Then we come up with our experiment. And it's, again, a very formal process. We do our experimentation. We're going to talk about, typically when we do that, we do a controlled experiment. And we'll talk about controlling for the variables in just a minute. And then we, again, come up with our results and then our conclusions based upon our results. And again, this can also be done in the field or the lab. So you can do the hypothetico-deductive model in both. So just to give you kind of an idea of the two types, the two ways in which we do science, induction and deduction, and how they're used, is this really simple example. It's straight out of your book, where you're going to go ahead, let's say you were doing a dive and you made some very specific observations. So in this case, you may have looked at all of these organisms and you said, okay, well, all of these organisms are fish and fishes. And fishes is a word, just so you know. So fish is, if we say fish and we're talking about multiple fish, they're all the same species. And if we say fishes and we're talking about multiple fish, then they're different species. So if you have a group of sailfish, let's say, that you're looking at, you would just say a group of fish. Even if there's, 10 sailfish, you would just say, oh, there's a group of fish. But if you have fish of different species, like you have here, then you would call them fishes, just as kind of fun fact for you. But you may say, okay, based on, your induction, you're looking at all of these fish and you notice that they all have gills. So based on induction, again, this is just one part of the scientific process, induction, where you make kind of these general conclusions on the basis of these observations that you made. You may come up with this general hypothesis that all fish have gills. All fishes have gills. So all fish species, all fishes have gills. And then you're going to say, okay, well, now I want to go ahead and take this step further and use deduction, which is where you're going to make very specific predictions, and you'll do an experiment for that. So again, here, you're going to maybe from this general hypothesis make the deduction that for your specific hypothesis that each type of fish has gills. So for each type of fish that you find, that fish will have gills. And so you're going to go ahead and do a bunch of testing. And in this case, it would just be making specific observations in all different marine environments and looking at all different marine fish. And you find that, lo and behold, all of the fish have gills. So then you can go back and accept your hypothesis. Woohoo, your hypothesis is accepted. All fishes have gills. But let's say instead, on this side here, that you made the specific observation that, okay, all of these organisms are marine organisms. You're a little more general. And then you also notice that all of them have gills. So based on your induction, you're going to say your very general hypothesis is all marine organisms have gills. Okay? And this is why we like to do that second process, the deduction process, where we come up with a more specific hypothesis to test that general one. So here in this case, if that's the case and all marine organisms have gills, then if you're looking at whales, whales should have gills. And of course, if you do the testing, lo and behold, you're going to find that whales have lungs and they do not have gills. And so then you would go back and you would go ahead and throw out kind of your hypothesis. So you would reject your hypothesis, and then come up with a new hypothesis. So again, that's the way in which we do science. And what's really important these days to understand is that when you're looking at data on the internet, please be very careful to remember that correlation does not always equal causation. And this is a really good example. This is just funny, but this is a really good example of an example where correlation does not equal causation. So again, here you can look and see that lo and behold, if you track ice cream sales, which is in blue here, and this is some actual data, believe it or not, and shark attacks here in red, you're going to find that they correlate with each other. And so, some people might say from this data that, oh my gosh, look, as ice cream sales go up, whoa, shark attacks start to go up. And so clearly, ice cream sales are causing more shark attacks. So obviously we know that's not the case, right? It's more likely that it just happens to be that ice cream sales increase with weather change when we have warmer weather, and that may be drawing in sharks for some reason, maybe mating, maybe feeding. Also, you're typically more likely just to have more shark interactions with people because during those warmer months there's just simply more people at the beach. So again, causation here, meaning, if you said, oh, well, increase in ice cream sales are causing shark attacks, right? That's not necessarily the case. So even though they're correlated here, correlation is not causation. So keep that in mind, especially again, when you're out there in the world looking at your own data. It's really important to understand that correlation is not always causation, okay? Really quick, I'm not going to spend a whole lot of time on this because you guys probably already know a hypothesis is just a simple statement about the world that might be true. Again, we haven't tested it yet. And most importantly, is testable and potentially falsifiable. Okay, so a testable hypothesis has to be one that's potentially falsifiable, one that at least potentially can be proved false. So this is funny because obviously, you can't prove this one false. So I just thought this was a very funny clip. I can't help but put in some fun cartoons every once in a while. So accepting hypothesis. Also in science, we don't like to use the word proven. Proven's kind of a bad word in science. And so no hypothesis can really ever be scientifically proven to be true. Instead, what we say is that these hypotheses are accepted for as long as they are supported by this available evidence that we have at this point in time, until some such evidence comes along later in life to show us otherwise, right? And then at that point, then we could reject the hypothesis. So, and this is the case with evolution and why we teach evolution in science. And the reason that we teach evolution by natural selection in science is that, again, this hypothesis and all of the evidence that we have up to this point supports evolution by natural selection. And so we accept this hypothesis, okay, until there is evidence to support otherwise. So again, that's a major thing to understand in terms of what science is and how we do science. That's one of the big bases of science, is this accepting a hypothesis. When we talk about experiments, oftentimes scientists have to create artificial situations to test a hypothesis. And this is because they can't control for and make the observations that they need under natural conditions. So a lot of times we'll have to, again, bring things into the laboratory. Again, funny cartoon for you here. And so your book has this example, but this is a very simple example of why we might bring, you might look and see mussels from a warm site and mussels from a cold site and wonder, that was your field observation, wonder why is it that you have the mussels in the cold site are smaller than the mussels in the warm site? And does that have to do with temperature? Is temperature a factor here? And so what you might do is, again, take the mussels back to the lab and go ahead and do a controlled experiment to see whether or not temperature was your factor for the different sizes of mussels. And so you can bring those mussels back, you'll just bring them back from the same site and expose them to different water temperatures, but keep all of the other factors, the same. So this is what a controlled experiment is, where you only allow one variable to be different, right? And then all of the other factors are the same. So the water quality would be the same, meaning you might have a shared pipe that splits and goes into each tank. So the water quality would be the same. The food would be the same. The amount of food would be the same. Again, the mussel species in this case would be the same. Potential disease would be the same. You would, maybe test water quality regularly to ensure that. And any other potential factors that you can account for, you would do, so that you would only vary temperature. So that way then you could accurately determine whether or not temperature was a factor. And so you could, maybe put a warmer in one tank and then the other tank wouldn't. And if, again, the mussels were the same size, then you could go back and say, okay, well, temperature wasn't the factor. But if the mussels were different sizes, then you could say that you could accept your hypothesis that temperature was a factor. So anyway, just a very simple way to show you what a controlled experiment is. Now, when is a hypothesis accepted as a theory? And a scientific theory is something very different than what we would, when we use the term theory in the general public, it means something very different than it does for science. So when a hypothesis has been so extensively tested that we actually consider it to be true by Occam's razor, this principle that, again, the simplest path is likely to be the one that is most true. And so that's when we accept something as a scientific theory. So when all of the evidence that we have in all of the experimentation that we have up to this point points to that hypothesis, then we elevate that hypothesis to a theory. And so again, this is why we have the theory of evolution by natural selection, because again, it's been something that's been so extensively tested, and we have so much data that supports that hypothesis. And so we accept that to be, at this point in time, true, until, again, there is other evidence that might show us otherwise, okay? But like any hypothesis, again, it is subject to rejection if evidence later on comes along and accumulates against it. So I love this image, because you're going to hear a lot, and I'm sure you have already, when we talk about even, the pandemic lately, we talk about looking at information that was published in peer-reviewed journals. And the reason that that's so important is because this is what it's like for those of us that have been through the process of publication. It feels like this, to get a paper accepted by a journal. And so you have all of these scientists, right, that are out there, and they are there with their knives and swords and whatnot to cut up your research and find any holes in your research that they can. And again, this is what gives science its strength, right? This is what makes science so valuable, is that we have this process of peer review, so that, when a paper is published, especially in a journal that's a well-respected journal, then we know that that is some good science. That was a good experiment designed to the best that we know at this point in time. So that's our foundation for science. There are, of course, limitations of the scientific method. Scientists are people too. We make, human error. Of course, we also have our own personal bias that comes into play. So, there are errors that can be made just because scientists are human too. Oftentimes, though, the nice part is that these errors are usually corrected by the fact that hypotheses are usually tested by multiple scientists and oftentimes multiple places. So that helps to give us some checks and balances for that limitation of science. Also, the other challenge with science is that it can't make judgments about values, ethics, morality. Again, that's outside of the limits of science. So I love this statement: science can reveal how the world is, but not how it should be. So it can tell you what's happening with climate change, but science can't tell us, science will not tell us what we should do with that information. That is up to us and policymakers to take that information and decide what we're going to do with that, and make those decisions based on that. So that's the tough part about science. It's challenging, especially in terms of marine science. So this all depends, again, on values, beliefs, and feelings that are beyond the scope of science. And that becomes challenging when you are studying marine organisms, because sometimes your feelings, right, might get into there, because we might have certain feelings about marine organisms and what should happen with them. Okay, so I think I'm going to go ahead and just really briefly talk again about the history of marine biology. I don't want to spend too much time on this, because this is just kind of a brief overview of how we got to where we are. So it's, of course, I think important to know at least who was considered to be kind of the first marine biologist. So Aristotle was considered to be our first marine biologist. He, again, was a 4th century BC Greek philosopher. So again, really really a man outside of his time, right? He actually did dissections and necropsies on animals. And so some of the descriptions of marine life that he made, we actually still use today. So we actually, he again did some dissections and discovered gills, and figured out that they were the breathing apparatus for fish. Again, I mean, how he did this in his time frame is just a little bit beyond amazing to me. So he was, again, a man well outside of his time. As we go through history and the history of marine biology, there are many different people that were major players. So we didn't talk about him, but James Cook is also, I'm just going to briefly talk about him. You don't have to know about him, but because you're going to hear about, schools, you're going to hear about marine schools named after him. He was this English captain, and he was one of the first captains to actually make scientific observations. He had full-time naturalists on his crew, and he was the first European to actually witness or observe Antarctic ice fields. And he saw Hawaii, he traveled to Hawaii and New Zealand and other Pacific islands and brought back all kinds of specimens of plants and animals. So he kind of was important in that he kind of started this interest in looking at marine organisms on these expeditions, in these ship crews. So, of course, by the time we hit the 19th century, it was a little more commonplace to bring a naturalist on board your expeditions. Darwin was one of the most famous, of course, of the shipboard naturalists. And he traveled on the HMS Beagle in 1831. And so, of course, you probably know him for his theories of evolution by natural selection and his theories of natural selection and adaptation. And so that's kind of what he's most known for. But and on his kind of expedition adventures, which are really interesting, he spent quite a bit of time on the HMS Beagle. And he actually got horribly seasick. But he stuck it out because, again, he had so much of a desire to learn about marine ecology and marine organisms. So he also made some really great contributions to marine biology. He actually explained the formation of atolls, which we're going to talk about, which are these rings of coral reef. And again, we use his explanations today. He collected all kinds of data on marine organisms in the southern oceans, which really hadn't been looked at because the southern oceans were very difficult to travel and on boats at that time. And so he collected some the first information that we had on marine organisms in the southern oceans. And then he actually developed these nets called bongo nets that were used to capture marine plankton. And we actually still use these bongo nets today we still call them bongo nets. They kind of look like a bongo drum here. But we still use these nets based upon his early designs of them to capture marine plankton today. So kind of interesting. Well, I'm not going to talk about the Wilkes expedition, although it was very important because in the U.S. in terms of bringing about exploration on expeditions in the U.S. But this was, for the U.S., kind of one of the most important voyages for exploration in the marine science field. It was their captain ended up being Lieutenant Charles Wilkes of the U.S. Navy, who actually was quite cruel and a very difficult person to have as captain. He actually promoted himself upon leaving port to captain, and ended up being court-martialed later for his mistreatment of his shipmates. But his expeditions were really important in terms of collecting information, especially flora and fauna, from all of these different islands, especially in the South Pacific and on the Pacific Northwest of North America as well. And so he ended up collecting something like, I think, 10,000 biological specimens, and over 2,000 of them were new species. So it was really an important expedition in terms of scientific discovery, but really a hard way to come about that. He was unfortunately not a kind person to have that happen. And then the last expedition that your book kind of talks about is the Challenger expedition, which was the first time that a ship, and they actually used an old British Navy ship, it was an old British Navy warship, was actually outfitted. They turned it into this kind of research expedition ship. So it was designed as with a scientific laboratory on board. The leader of it was Charles Wyville Thompson, and this was the first time that they had a real scientific expedition, where it was an expedition for the purpose of conducting science. They were surveying the oceans, collecting water and sediment samples, of course, doing all kinds of biological samples. And they accumulated so much data from this expedition that it took them 19 years to actually publish all of it. And it ended up being in like 50 volumes. So the Challenger, that was called the Challenger expedition. It was really an important expedition. Sorry, I just always like to give you a little bit of the history, important information on how marine biology came about. So submarine warfare was really important. So that was kind of the next big impact in history on marine biology was World War II. And in World War II, submarine warfare was obviously a big part of World War II. And so that was important in terms of marine biology because this led to the discovery of sonar. And so sonar just stands for sound navigation ranging, in case you're on Jeopardy one day. And it's just based on the detection of underwater echoes, right? And this was really important because at the time, it was very difficult to use sonar because there was a lot of what we call noise, meaning that the oceans were alive with other organisms. And how did you figure out what was an organism, right? Like a big whale versus a submarine. And so they ended up developing these labs to be able to identify this animal noise so they could kind of cancel it out and use the sonar for what their intended purposes were. And so they ended up developing labs such as the Scripps Lab. So this was, again, really important because, again, the Scripps Lab ended up eventually becoming a very important marine laboratory, marine discovery center after World War II and after, again, its contributions to submarine technology. So again, this is sonar. You guys are all familiar with sonar, emitting at ping, and then again, taking back in that echo and measuring the time frame it takes, and then again uses algorithms to calculations to estimate depth. And they ended up using this, right, sonar, to map the ocean floors. So when we look at some of the topography of the ocean floors, again, that information is based mostly off of sonar. In the years shortly after World War II came the development of my favorite, which is scuba, of course. Thanks Gagnon and Cousteau, who were the ones that came up with scuba. And of course, your Jeopardy question here is, what does scuba stand for? And that one's an easy one. Probably most of you know that. Self-contained underwater breathing apparatus. And the amazing part about scuba, of course, is this allowed scientists for the first time to observe marine organisms in their natural habitat. We could get this, what we call life history information on them, because we could observe organisms throughout their life cycle. We could see, what was happening, where they laid their eggs, and how they did that, and what happened after the eggs hatched. And so again, we could get all of this awesome life history information by using scuba to make these types of observations. This one's a really interesting photo because this is really old. The scuba equipment today is a little bit more streamlined. And so again, scuba allowed observations of marine organisms finally in their natural habitats. So marine biology today. Obviously technology has advanced marine biology greatly in the present day. And so research vessels are roomier, they're equipped with, the latest navigation, the top technological research equipment, and they're much, much more high tech. So this is a very cool image of what's called FLIP. This is the floating instrument platform that Scripps still uses today. And please, I know I'm running out of time. I don't want to make this video too long, but please watch these videos on FLIP. It's really interesting because the videos will show you how it goes from this kind of horizontal platform and then flipping up to the vertical platform. It's cool because it shows you on the inside of the ship, you can see how they have a lot of their equipment on trundles. So you see like coffee pots and things like that that are sitting there on these trundles, and as it flips, the equipment and the platforms that they're on will move with it so that the coffee pot stays upright. So again, really, really kind of interesting. And then you can see some of these platforms. The last video that I have down here, the walking tour, actually takes you, and that video is a more recent video. So it's pretty cool. And it takes you out on this platform. So again, this was a really, really interesting, again, development for research vessel by Scripps, because you can, again, spend weeks out on this platform in the middle of the ocean and gather all kinds of interesting information. We also, of course, use deep sea submarines today, remotely operated vehicles where we can send down these vehicles especially into these really deep environments that are remotely operated from the surface. Autonomous underwater vehicles, so some of them are self-driving and are programmed to do so on their own. Also, we have underwater marine laboratories. So Aquarius is one of the big ones here in the Florida Keys. And especially researchers, we actually have someone on campus. If you know Dr. Churosky, he was actually an aquanaut at Aquarius. So you can go and stay at Aquarius underwater. And I believe it's about like 70 feet down. It's about 70 feet down, and you can stay there for a week or 2 weeks and do research, and that's a huge advantage for you to be able to just literally walk out the door and scuba dive and do research, every day. So really interesting. And then, of course, we use marine mammals today. So we'll use marine animals as kind of autonomous samplers. We can put cameras on them to actually gather all kinds of life history information about them. Also gather information about the surrounding environment, what's happening in the marine environment that they're in as well. So, and here's also another short video clip if you'd like to watch on that. And then, of course, we now have educational institutions that take out students and train students on marine biology as well. And then computers. I love this image because these computers are so old. So I've kept it in here all these years. It's great. But obviously computers have had a large impact on marine biology and marine biology studies. Especially our computers today, those are quite old. But computers today, not only impact how we process information and use that information, but how we share that information as well. And of course, we have satellites today. So satellite imagery gives us quite a bit of information, especially when we're talking about something like remote sensing, where we're using technology to study the Earth from a distance. So we can use that. In this case, this is kind of a cool image where we used satellites to show the abundance of photosynthetic organisms. And so you can see as you go from kind of purple, this is phytoplankton, the pigment concentration. And as you go down towards the red, you see you have a higher concentration of photosynthetic organisms. So kind of interesting to see the distribution of photosynthetic organisms, planktonic photosynthetic organisms. So anyway, just kind of interesting information on how we study. Please make sure that you read the Ion Science Carbonate Experiments on the Reef. It's a very interesting article talking about carbon dioxide in our oceans and a little bit about acidification. So please read that on page 14, or you can click on the link to the PDF of the article that I have posted in Canvas. All right, so I will see you back here for the next lecture.